My name is Rachel Rubenstein, and I'm a technical product manager here at Oxford Nanopore Technologies. So I'm excited to take you through some product updates today and tomorrow to give you a bit of an overview of what's new and talk about some updates. Today's talk is going to focus on human genomics, and we're excited to show you what our products are capable of there. Today we'll be going over various applications from targeted sequencing, including CNVs, SVs, and methylation, as well as whole genome de novo assembly, and some options for sample prep and data analysis. Also, at the end, you'll hear from a very special guest about his research and how he applies these technologies in his day-to-day life. So, without further ado, let us get into the human genomics landscape. So the optimal way to unlock insights into the human genome is through the PromethION line of devices. From the PromethION P2 Solo through the PromethION P48, the PromethION line of sequencing is more accessible than ever. And all of these devices take advantage of our PromethION flow cell, which can produce up to 200 gigabases of data per run. So if you are really excited, in 1 year, you could sequence 5,000 human genomes at 30x coverage with 1 genome per flow cell, or 10,000 genomes at 15x coverage using 2 genomes per flow cell. And that is a lot of data, so I'm sure you'll have a lot to do no matter how many genomes you sequence. And as we all know here at Oxford Nanopore, great data starts with great DNA extraction. On the Nanopore community, you'll find various extraction protocols as well as tips and tricks, along with recommendations from other users and customers on how to optimize your extraction. Additionally, we publish a few preparation protocols, including a protocol that provides N50s of 10 kilobases, which is great for high coverage of large genomes, and an all-new protocol that provides N50s of 30 kilobases, which uses our Kit 14 chemistry. If you're interested in ultra-long sequencing, our customer record so far is 4 megabases in a single read, but hopefully one of you will break that. And with that protocol, you get N50s over 50 kb, giving you telomere to telomere insight, which hopefully one day will help us resolve a gapless human genome. Throughout the prep process, you have the opportunity to either zoom in on your sample using adaptive sampling, or get a bird's eye view with whole genome sequencing. You can either focus on variation or do de novo assembly. So you just heard a lot about adaptive sampling in the previous session, but just for people who might not have heard, adaptive sampling allows you to have finer control over your sequencing by specifying targets to either enrich for or deplete. Adaptive sampling is completely software controlled, meaning that you do not have to do any additional upstream prep or enrichment to get the benefits. This method is unique to us, and as you can see in the graph, compared to the control, you get much, much more coverage on your adaptive sampling targets. And here you can see that compared to the control, you get really great coverage across all 202 targets there. This method will open the doors for human genome research because you can target one or many chromosomes at the same time, and you get methylation info retained throughout this process, so you can use that data to paint a complete picture. However, if you're more interested in whole genome analysis, we have you covered there. With our new Kit 14 chemistry, we give you the ability to do both simplex and duplex reads, giving you adaptable sequencing control with accuracies of 99% and reads that are over Q20. With simplex reads, you get highly accurate, high-throughput reads that are the perfect tool for CNVs, SVs, and methylation analysis with applications that range from human disease to cancer research and much more. With duplex reads, accuracy is paramount, and you'll get Q30 reads, which you can rely on, that also supports telomere to telomere research and whole genome assembly. It is good to note that duplex reads are compute intensive, so it's good to have a specific goal in mind before going down this route. Luckily, the high simplex accuracy means that you'll get useful data no matter what you're interested in. Now, we also cannot talk about end-to-end analysis with end-to-end protocols without mentioning analysis. So For analysis of human genomic data, we have a human variation workflow that is available on the command line or through the Epitome Labs interface. This workflow is Nextflow-based, and it calls SNPs, SVs, and reports methylation. If you want to learn more about this workflow, please find time during the break to go downstairs to the Data Analysis Live Lounge, because they're really excited to talk to you there. And on the screen, you can see just a snippet of the human variation workflow, which can run on the devices. So while I could talk to you all day about how great our technology is and how great it is for human genome research, it would be way better for you to hear it from our special guest expert. So now I'd like to introduce our guest, Dr. Danny Miller. And Dr. Miller is the PI of the Miller Lab at the University of Washington in the US. And his research goal is to make genetic disorder diagnosis more efficient and effective. Additionally, he wants to understand disease through human genomic variation, and he is the leader of the 1,000 Genomes ONT Sequencing Consortium. So please give a warm welcome to Dr. Danny Miller.
So our lab is interested in shortening the amount of time it takes to make a genetic diagnosis, increasing the rate of genetic diagnoses, and also decreasing barriers to getting a comprehensive genetic evaluation. I'm a clinical geneticist. I see kids in skeletal dysplasia, general genetics, and I can tell you, unfortunately, that there's families who prioritize going to one visit over another. And oftentimes they choose like the neurology visit over the genetics visit because they just can't get the time off work, they can't get to the hospital, things like that. So we want to take away those barriers and help those kids get comprehensive evaluations.
Awesome. Just about those barriers, so are those mostly access to appointments, or is it technological, a little bit of both?
It's often just getting time off work and just making it to appointments, very simple things. Or navigating the insurance process in the US, unfortunately. So our goal is a single test, analyzed in a stepwise fashion, that doesn't need to be reauthorized every time, right?
Cool.
So you could just say, I want to look for copy number variants. If it's negative, I want to look for SNPs or indels and genes of interest. If that's negative, I want to do an exome. If that's negative, I'll do the whole genome.
Awesome. And obviously, you've been using Nanopore Tech for that. So what's been the most interesting piece of data or insight that our technology has helped you uncover?
That's a hard question to answer. It's hard because we've done so many different things. Just as an example, we just posted a preprint. We did an assembly of a little mouse called the African spiny mouse. It's an interesting critter because you can rip some of its skin off and it'll grow back perfectly, scarless. You can infarct its kidney. It'll regenerate, or the kidney will function again after time. So our nephrologists, for example, are very interested in what is it about that mouse that makes it able to do that. So we did a nearly complete assembly of that mouse, which allowed us to do improved RNA-seq analysis, improved variant calling, things like that. But honestly, I think I would be remiss if I didn't say that one of the best things is when we find a variant missed by standard clinical testing for a family, and we can either explain, a child's, symptoms or phenotype, and either give the family the extra information that they couldn't get from standard clinical testing. We had a case we looked at last night, actually. Translocation, found the exact breakpoints, and it kind of explains everything about the child's phenotype, and it really kind of answers a lot of questions for the family.
Got it. And what enables you to see the things that are traditionally missed? What do you think that, I guess, aha moment is?
It's honestly, I think, how much simpler having long reads makes doing these analyses. With short read data, say you're looking for a copy number variant, and everybody knows the variability you get with short read sequencing. With long reads, it's really nice and smooth. So when you see those kinds of structural changes, they just kind of stand out at you. The structural variant callers are also much more sensitive with long read data, right? We can detect 25, 30,000 structural variants per person with long reads. You can detect maybe 10, 15,000 with short reads. So you get a lot more sensitivity that way.
Sweet. That sounds really great. Speaking of all that and detection and all these different variants you can call, do you mind talking a little bit more about your work with the 1,000 Genomes Consortium?
Sure. Yeah. One problem when we long read sequence someone is that we find 25 or 30,000 structural variants. And how do you prioritize those? Which ones are common? Which ones do we share? And which ones are unique? How many transposable element insertions near a gene do we all share? Or tandem repeats, for example? What's the distribution in the population? So we need a truth set for structural variants. Our goal is to sequence actually all 3,000 of the 1,000 Genomes Project samples. We're starting with 500 of those samples now to show that we can, to get a sense of what the allele frequency is down to 1%. use that for filtering, use it for calling variants in genes that are difficult to analyze with short reads. 400 to 500 genes or so are difficult to analyze with short read sequencing. Some of these are medically relevant genes. So what's the variation look like in those genes in the population? Our goals are to generate about 30x coverage per sample with read N50s of greater than 50 kb or so. We do haplotype resolved assemblies, we call variation, and we share the data. So you could actually go online right now and download some of the data today.
Sweet. And I'm going to put you on the spot a little bit. Right before this, you were telling me about your N50s and your super long reads that you've been using with adaptive sampling. So do you mind sharing?
Yeah, of course. So we did a lot of work with adaptive sampling before it was in Minnow, of course, when it was still reedfish, things like that. We've used it now on the Promethean, and we absolutely love it. We love it because we don't shear the DNA, so we can target specific regions and pull out reads that are, I mean, literally 300 kb reads. We had a family with an X chromosome variant and near a structural variant, so they had an inversion and deletion. And to fully resolve that region, we needed reads that were over 100 or so kb. We got pretty close just with whole genome sequencing, but we wondered, could we use adaptive sampling to really increase the coverage of long reads in that region? And it worked great. We got 40 or 50x coverage with reads, well over 100 kb.
And what sample types are you starting from?
Typically blood. Sometimes we'll do DNA derived from tissue. As everyone knows, FFP is bad or it doesn't work well. Saliva or buccal swabs actually work pretty well if you're careful about how you extract them. So we've worked, I think, with most things at this point.
Cool. Thank you. Well, before I, ask Danny questions to death, do we have any questions from the audience? Just please wait for the microphone. Awesome.
So you talked about the super ultra long 150 kb.
I'm sorry, we talked about what?
You talked about using no fragmentation, no shearing of your DNA, and to get the 150 kb beyond the super ultra long read for adaptive.
Right.
How that affects your cost?
What's the cost of that?
Yes.
Are you asking how does the cost change with super long reads and no shearing?
No, so real numbers in a way.
Yeah, so
for clinical application, it becomes very important, I think.
Yeah, for that, it's the cost of the flow cell and the library prep. So
I needed some specific numbers. For example, 150 kb, that project, really.
So it depends on, it's all a function of how many reagents you buy, of course, right? So, on average, it probably costs us about $1,200 US dollars per sample. So for that adaptive sequencing run, it probably cost us about $1,200. So single flow cell, Promethean flow cell, one library prep that we load, run for 24 hours, wash, run again. That one we only did 2 loads, so we were able to save the flow cell as well and use it for something else later.
Cool.
When you do 150K adaptive, what is the level of enrichment?
Yeah, so that's going to depend on your average fragment size. For that
you
particular,
said that actually ultra super long, 150K and 50.
Yeah, so for that particular experiment, we got about 50x coverage of the region that we were interested in. And we targeted 37 megabases in total in that particular experiment.
Any other questions? No? Well, I guess I'll ask another one. What has been, I guess, the greatest learning moment? I'm trying to phrase it positively. What is one mistake you've learned from that you could share with everyone else?
Always had the ligase when you're doing the ligation protocol. That's actually why I'm not allowed in the lab anymore. I think having good quality sample go into your experiment is going to help drive the quality of your experiment. So know what you're bringing in, and that way you won't be disappointed with the results you get out. I think that's something that it took me personally a long time to really figure that out, and now we've got that set really nicely in the lab. So yeah.
Nice, thank you. All right, one more opportunity for questions. If not, you can find us both throughout the conference. So just before we end, I want to summarize what we've been over. So today we talked about the capabilities of Promethean flow cell and the Promethean line of devices from our favorite P2 Solo all the way to the P48. We talked about the end-to-end protocols available on the Nanopore community, the benefits of both simplex and duplex reads, as well as the benefits of the data analysis that you can do with the human variation workflow. So I want to thank Dr. Miller again